The overall goal of this procedure is to evaluate transport of a drug delivery system or the therapeutic agent it carries across a cellular barrier, such as a model of the gastrointestinal epithelium relevant to oral drug delivery. This is accomplished by first culturing Keiko2 cells on transwell inserts to allow cell differentiation and formation of a cell monolayer. The second step is to validate that this model represents a permeability barrier by measuring the transepithelial electrical resistance, immunostaining cell junctions, and assessing passive permeability of a detectable tracer molecule. The next step is to prepare the drug delivery system. Targeted polymer nanocarriers are used in this example. Carriers may contain a drug cargo or not, and controls such as non-targeted carriers or a free drug must be included. These elements are labeled with radioisotopes to allow their detection. The carriers are then added to the apical chamber above the cell monolayer. After incubation for varying time points, carriers are collected from the apical chamber, the basolateral chamber, and the cell monolayer, and tested for potential degradation of the labeled component. Nanocarrier transport across the cell monolayer is then calculated. Finally, to investigate the mechanism of transport, similar experiments must be conducted in the presence of pharmacological inhibitors of transcytosis across cells or with reporters of paracellular transport between cells.
The technique we describe here is an adaptation of well-established methods used to evaluate the rate and mechanism of transport of drugs and drug nanocarriers across cell monolayers in culture.